Dear students, so now we will see the topic of ANDA, abbreviated new drug application. First, before going into this, we'll discuss the previous topics, that is IND and NDA. What is IND? Investigational new drug is a submission to FDA requesting permission to initiate a clinical study of new drug product, and it's also a goal to determine if the product which we will be studying is reasonably safe for initial use in human. What are types of IND? Investigator IND, emergency use IND, and treatment IND. Okay, next is new drug application. It is the data which contains all the information that the FDA requires for approve a new pharmaceutical for sale and marketing in the US. Also, the application to market a new drug, biologic or antibiotic drug for human use. So thus the related application is known as new drug application. So now we will be studying ANDA. After new drug application is for a patented drug. Okay, after the patent expires and when it goes to generic product, thus ANDA filing is required. So generic drug application are referred to as abbreviated new drug application. Pharmaceutical companies must admit ANDAs and receive FDA's approval before marketing new generic drug according to 21 CFR 314-105D. Once ANDA is approved, an applicant can manufacture and market generic drug to provide safe, effective and low-cost alternative of innovator drug product to the public. Once the ANDA is filed and approved, the applicant or manufacturer can market a generic drug. So once the ANDA is filed and approved, the applicant or manufacturer can manufacture a generic drug or generic product and market the same product to provide a safe, effective and low cost. But it must be bioequivalence. The product which will be manufactured and marketed must be bioequivalence with the patented or innovator or standard drug. Generic drugs are termed abbreviated as they are not required to include preclinical and clinical data to establish safety and efficacy. Why it is called abbreviated? Abbreviated means same, comparable. So why it is called abbreviated? Because it does not include the preclinical and clinical data, means preclinical or which is studied on animals, or then after later if it is safe and efficacy, it is further studied on human beings. So these studies are not required to manufacture a generic drug. Only the requirement is the it should be bioequivalence, means the pharmacodynamic and pharmacokinetic studies should be same. Okay, they must scientifically demonstrate bioequivalence to the innovator or brand name drug. A generic drug is comparable to innovator drug dosage form, strength, route of administration, quality, performance and intended use. A generic drug, as we are manufacturing or producing, must be comparable to the standard drug with its dosage form, strength should be same, label claim should be same, its route of administration should not be changed. The quality which the standard reference drug shows, the same quality should be shown or must be in your generic drug. The performance means bioavailability should be same as that of standard, and the use. Okay, one of the ways to demonstrate bioequivalence is to measure the time taken by generic drug to reach bloodstream in 24 to 36 healthy volunteers. The time and amount of active ingredient in bloodstream should be comparable to those of innovator drugs. So, thus to demonstrate, or when we will show that bioequivalence is same as that of innovator drug. Thus, after intake of the drug or product, the active pharmaceutical drug which is absorbed by the body and reaches the site of action. So the samples, the blood samples should be taken in every 4 hours or every 6 hours till 24 to 36 hours. Okay. And when a standard or innovator drug is given to a person, its body samples should be taken up to 24 to 36 hours, and another person who consumes the generic drug should also be sampled simultaneously or at the same time. So by simultaneous sampling of both the human bodies, which one has consumed the innovator drug and other has consumed the generic drug. So how we can show or demonstrate a bioequivalence in a healthy volunteers? Generally, what is done, that if we want a bioequivalence studies, that its bioavailability should be same. Example, two persons, person A and person B. Person A is given the standard or patented or innovator drug, and person B is given the generic drug, okay, of the same active product or same active drug. They consume a formulation or a product at the same time, and they are monitored up to 24 or up to 26 or 48 hours. Their blood samples are taken after every 4 to 6 hours at same time. Okay, so that when we take these blood samples at the same time, their studies of the innovator drug and generic drug will match or will show the results of the same time. Use of bioequivalence as base for approving generic drug products was established in 1984, also known as Waxman-Hatch Act or Hatch-Waxman Act. It is because of this act that the generic drugs are cheaper without conducting costly and duplicate clinical trials. As earlier, before 1984, there was no act for producing or generating the generic drugs. So Hatch-Waxman Act brought this ANDA filing, and generic drugs were then being manufactured. Format and content of ANDA. There should be 3 copies of the ANDA for submission. The first should be archival copy, it is the permanent copy, a review copy, and a field copy. An archival copy shall contain application form, table of contents, basis for ANDA submission, conditions for use, active ingredients, route of administration, dosage form and strength, bioequivalence and bioavailability, labeling, chemistry, manufacturing and control samples, patent certification, financial certification or disclosed statement, other information, and the form which is filled comes under the section of 314.94 A12. The patent certification includes one of the following. The first is the paragraph 1 certification that the patent information has not been submitted to the FDA. If any patent whose information has not been submitted to the FDA comes under the paragraph 1 certification. Second paragraph is such patent that has been expired. Third paragraph is the patent will expire, means such that its expiry of the patent is coming soon, comes under the paragraph 3. And paragraph 4 certification is that the patent is invalid, unenforceably, or will not be infringed by manufacturer, use or sale of generic drug. It is such that when a applicant or manufacturer files a duplicate copy or some false statement or false matter or false results, that which after review comes under and is notified, and they are not permitted to manufacture, use or sell for the generic product. This is the ANDA chart. First, the applicant applies or files the ANDA. After ANDA filing, it goes through the review board. During ANDA filing, the concerned department shows or accepts or denies, which refuses the file which is issued. If it is refused, the applicant again edits or make changes and applies again. If the ANDA file is accepted, it is reviewed by the OGD CDER, that is bioequivalence study is reviewed, chemistry or micro is reviewed, then after request for plant inspection. After the review is completed, the department requests for plant inspection, means they comes to the site where the product is being manufactured or study is being carried out, and also labeling review. Labeling review means label claim which we have given on the label. Then bioequivalence review acceptable, and chemistry micro labeling review acceptable. If if the bioequivalence study, which is reviewed bioequivalence or chemistry micro review, is completed and if it is accepted, the pre-approval inspection is done. Pre-approval inspection means before approval of the product. The committee against or FDA against visits the site and they audit the site. Okay, if bioequivalence, after review of bioequivalence, if there is any deficiency, the deficiency letter is given and the deficiency letter is also given to the sponsor. Okay, then approval deferred pending satisfactory result. If the pre-inspection is not satisfactory, then the approval team which comes for inspection, they give a pending points or deficiency points, and the site again they fulfill the requirements, and after the requirements are fulfilled and there is no any deficiency or pending document, then ANDA is approved. Then after the ANDA approval, the manufacturer or applicant can go for this sale, use and market the generic product. And thus conclusion, what is NDA? What is difference between NDA and ANDA? NDA is applicable for a new drug, that is to make a new formulation with a new drug, or okay, ANDA is applicable for generic drug. After a new drug, when patent ends, it comes to the ANDA, it comes to the generic. ANDA takes about 12 to 15 years as they go through the preclinical studies, clinical trial studies, phase 1, phase 2, phase 3. Thus they require a long time as compared to ANDA. As compared to NDA, ANDA requires only 1 to 2 years as they need only a bioequivalence study to be done. NDA is more expenditure of money. Comparatively, ANDA is less expensive. The cost of drugs are more. Why the cost of drugs of the patented drugs are more? As they take longer time and they do the clinical, preclinical studies, phase, various phases. Several times the tests are done. The manpower, the utility is required in 12 to 15 years is a very big time. Thus, it is the cost consuming. Okay, comparatively, generic drug or ANDA are very less. Non-clinical studies and clinical investigations are essential in NDA. As it is a new drug or new formulation, the non-clinical or preclinical or clinical investigations are necessary. Whereas ANDA only requires the bioequivalence and bioavailability study with compared to the patented drugs. Exclusivity. Exclusivity is a statutory provision designed to promote a balance between an innovator and generic drug competitor. Okay, it is a balance between an innovator. The time required between the generic drug and the patented drug is known as the exclusivity period. As long as the drug patent lasts, a referred listed drug company enjoys a period of market exclusivity or monopoly. Expiration of patent removes the monopoly of the patent holder. Terms of exclusivity. Orphan drugs exclusivity is for 7 years. Any new chemical entity is for 5 years. Any pediatric exclusivity is for 6 months. Patent challenge is for 180 days. Hatch-Waxman amendments and 180-day exclusivity. Before Hatch-Waxman amendment, generic manufacturer could file ANDA only after innovator's patent expiry on cancellation. But under section 505J 5P of Hatch-Waxman amendment, it permits preparation and filing of ANDA before patent expiration, so that the effective approval date of generic drug would be on expiration date of the patent of the innovator original drug. The Act also establishes another procedure in which the generic company can challenge patent of the innovator. For generic companies, the amendment provides an inventive 180 days exclusivity period in which no other ANDA for that drug can be approved. This 180 day period is to encourage generic companies to challenge validity of Orange Book listed patents or to design around these patents to bring more quickly a generic drug to market. For innovator company, filing of an ANDA is an act of patent infringement. So if innovator company brings suit within 45 days, the approval of generic company's ANDA is delayed for up to 30 months. Waxman-Hatch amendments benefit to innovator companies 45 days to claim. After that, if it suits, then it is delayed for 30 months. If does not suit, the generic drug company can manufacture and they can take benefit of 180-day exclusivity period and also to challenge the patent drug. Okay, thank you. This is all. So that we have studied IND, NDA and ANDA. Okay, thank you.